And now I'd like to welcome Lindsay Cowell from UT Southwestern to speak to us on statistical classifiers for detecting phenotype-associated biophysicochemical motifs in air. Thanks, Lindsay.
Yeah, I'd like to thank the meeting organizers for inviting me to speak. It's really exciting for me to speak to this group about my research rather than all this repository stuff that we usually talk about, which is really important, but the only reason for those is to do this kind of thing. So thank you for the invitation to speak. So one of the primary goals in my research group is to identify patterns in immune repertoires that are associated with host phenotypes, like a lot of people in this group. So far, we've worked in two application areas, cancer and autoimmune disease. So in the context of cancer, our goal has been to identify tumor-associated T-cell receptors. And in the case of autoimmune disease, our goal has been to identify immunoglobulin sequences that are associated with relapsing, remitting multiple sclerosis. So first, I'm going to go through the computational approach that we are using to identify these sequences, and then I'll present results from colorectal cancer, breast cancer, ovarian cancer, and then also multiple sclerosis, time permitting. And then I'll briefly introduce our plans for follow-up studies. So the literature is full of studies with similar goals. The vast majority of them have applied the typical methods that really utilize only a tiny bit of the information that's embedded in immune repertoires. So calculating repertoire level summary statistics such as diversity and clonality, restricting the set of sequences analyzed to those from highly expanded clones, or looking for amino acid sequences that are shared across individuals with the same phenotype. And these are valuable approaches that give us important information, so I'm not criticizing them at all, but I think they barely scratch the surface of the amount of information available, so we don't want to just stop there. So one of the things in my group that we were interested in doing was coming up with approaches to mine the full set of sequences from all samples that we're analyzing, rather than just looking at repertoire level summary statistics or highly expanded clones. And then also to use, to convert from an amino acid sequence representation of the receptors to a representation that captures important features of the receptor's antigen binding properties or potential. And so in the first set of examples that I'll show from cancer, our specific question was, can tumor-infiltrating lymphocyte TCR repertoires be distinguished from the repertoires of lymphocyte populations that are resident in healthy tissue of the same organ type on the basis of the antigen binding properties within those TCR sequences? So for colorectal cancer and breast cancer, the first examples I'll show, we used publicly available data. This shows the study design for the original studies. The sequencing was done by Adaptive Biotechnologies, and they made the sequences publicly available in their immune access database. So in those studies, they started with patients. From every patient, 2 samples were collected, 1 tumor sample and 1 adjacent healthy tissue sample. And then deep TCR sequencing was conducted on all samples. And our goal is to utilize the CDR3 sequences within those repertoires to predict for each repertoire whether it was derived from tumor tissue or healthy tissue. Or put another way, to identify the individual TCR sequences within a repertoire that mark that repertoire as being tumor-derived. And our hypothesis, for which we have no evidence, but our hypothesis is that such sequences may be interacting with tumor-associated antigens. Okay, so to do this, we first extracted all of the amino acid sequences of length 4 from the CDR3 sequences, all CDR3 sequences and all repertoires. And throughout the talk, I'll refer to these as snippets or formiers. We then convert each snippet from its amino acid sequence representation into a physicochemical representation using actually factors. And we then apply leave one out cross-validation to develop a model that will allow us to predict a label at the level of the sample from the set of all snippets or formiers that are within that sample. And so to do that, we score each snippet and then we aggregate the scores to assign the sample level label. So that was an overview of the approach. So now I'll go through it quickly in detail. So first, why formiers? So that was determined by analyzing crystallographic structures. So here you can see the structure of a T cell receptor in complex with its cognate antigen, so peptide bound to MHC. The MHC is whited out here, so you can't see it, but the antigen is shown here in red, and the CDR3 sequence of the T cell receptor is shown here in green. And an analysis of the structure revealed the CDR3 residues that were in direct contact with peptide. And you can see those highlighted here in blue, and they lie basically in a contiguous strip. So we analyzed 70-some structures, and their CDR3 sequences are aligned here, and the amino acid residues in each one that was in direct contact with peptide are highlighted in red. And the bar chart here shows the number of structures for which the residue at that particular position was in contact. And you can see that for a high proportion of them, a contiguous strip of about 4 amino acids were in contact. So we used 4. So next we extract formiers from all CDR3 of all sequences and all repertoires. And again, our hypothesis is that for the tumor-associated repertoires, for each repertoire, at least one of these formiers will be in direct contact with peptide for a tumor-associated antigen. Okay, so next we convert each snippet from its amino acid sequence representation to the physicochemical representation. And that is important because it allows us to capture physicochemical similarities between distinct amino acid sequences that may allow them to interact with the same antigen. So for example, both arginine and lysine have positively charged side chains. So to do that, we used Ashley factors. Ashley et al. Started with about 50 amino acid properties, and they applied dimensionality reduction techniques to come up with a set of 5 factors that maximize the amount of information contained while reducing the amount of redundancy. And those 5 factors correspond loosely to these properties. So take, for example, this snippet, arginine, tyrosine, arginine, aspartic acid. So for each snippet, we create a numerical vector by taking the 5 Ashley factor values for each amino acid within the snippet, and that results in a vector of length 20. So now what we have is for each sample, we have a bag of snippets, but in Ashley factor space rather than in amino acid sequence space. And our goal is to predict a label at the level of the sample or bag on the basis of the set of snippets that are found within that bag or that sample. So to do this, we used a machine learning framework referred to as multiple instance learning, which was designed to predict labels for sets of objects on the basis of the individual objects within the set. So to do that, again, we score each snippet within the set, and then we aggregate the scores to assign the sample level label. So to score each snippet, we used a linear combination of its Ashley factor values represented here by F, each with its own weight, represented by W. And then we use also an offset term that basically sets the threshold score above which a snippet would have to score in order to mark its repertoire as being tumor associated. What's not shown here, this is what happens when you make slides at 4 a.m. on severe jet lag. There's another term here which I left out, which captures the relative abundance of the snippet within the sample. So then we aggregate all the scores. We use a function to aggregate all the scores from a sample and then calculate the probability that the sample is tumor associated given that set of snippets or set of scores. For the results I'll present today, we just simply took the maximum score under the assumption that snippets in direct contact with a relevant antigen are rare within the sample. And then to calculate the probability here, we just used a logistic function. So the next step is to determine the values for all the weights that will maximize the probability of calculating correct labels, predicting correct labels. So to do that, we use a standard leave one out cross validation. So we start with N patients, each with a healthy sample and tumor sample. We exclude both samples from the same patient, and then we use the remaining 2 times N-1 samples for parameter fitting. We randomly initialize the weights. We then apply gradient optimization techniques, iterate 2,500 times, and then after going through that once, we go back up, initialize starting values again, and we do that a total of 250,000 times. Then from among those 250,000 models, we select the one with the lowest error in terms of predicting labels for the 2 times N-1 patients, and then we use that best model to predict labels for the held out samples. And then we put those back in and hold out the next 2 samples and go through that for all the samples in the study. Okay, so now for some results. So for colorectal cancer, as I mentioned, we use publicly available data. This was the original publication, and the data are available here from Immune Access. This just gives a snapshot of the patient characteristics here. Okay, and so here are our results. So here on the x-axis is indicated patients. So there were 14 patients in this study. On the y-axis, we show the probability calculated by the model that a particular sample was tumor associated. The red diamonds indicate the tumor associated samples, and the green circles indicate the healthy adjacent mucosa samples. And so you can see, so here this one and this one are quite close to the threshold, so the model has not very much confidence in its prediction, but they are technically correctly classified. Both probabilities were above 0.5. So all the tumor associated samples were correctly classified. 2 of the 14 healthy mucosa samples were incorrectly classified. I should have mentioned that the 2 samples from a single patient are scored independently, so the model has no knowledge, even though they're simultaneously held out, that information that they're both from the same patient and that one has to be tumor and one has to be healthy is not utilized by the model, so the labels are independently predicted. And so this gave us a classification accuracy of, I think, about 93%. So this shows the model weights. So each sort of block in the figure corresponds to one actually factor value, and then here the relative abundance of the snippet. And then each individual arrow corresponds to that position within the snippet. So like this position 1, this corresponds to the first amino acid in the snippet, the second amino acid in the snippet, third and fourth. So basically what you can see here is the model predicts a high probability of a snippet being tumor derived if it has a lot of small positively charged amino acids that tend to appear in alpha helices with 2 hydrophobic or let me say it this way, 2 hydrophilic amino acids flanked by 2 hydrophobic amino acids, and where the snippet is overrepresented in tumor tissue relative to healthy tissue. This shows the CDR3 sequences from each patient that received a high score. And the snippet from that CDR3 sequence that received a high score is highlighted here in red. And it's interesting to note here shows the rank of the abundance of that of receptor containing that CDR3 sequence within the sample. And interestingly, in many cases, we're not getting highly abundant TCRs derived from highly abundant clones. Some patients have multiple sequences or sequences that contain multiple high-scoring snippets, which is kind of interesting. Okay, so now here's the breast cancer study. Original publication, again, the data from Immune Access, snapshot of patient characteristics, so a variety of cancer types. And here are the results, same layout as before. Similar results also in that all the tumor-associated samples are correctly classified, although for some of them the model's confidence is low. And we have, I think, again, 2 healthy samples that were misclassified, but in each case the tumor-associated sample did have a much higher score than the healthy sample from the same patient. And here shows, again, the model weights, but I'll just move here so you can see that they are different from what we got for the colorectal cancer results. So the biophysicochemical motif that the model is keying in on is different for the 2 cancer types. And in this case, we are keying in on receptors that are among highly expanded clones. So that was an interesting difference between the 2. So in the case of ovarian cancer, this was sequencing conducted on patients from my institution, UT Southwestern Medical Center. So in this case, the study design was a little bit different, where instead of having healthy and tumor tissue from the same patient, we actually had patients who are cancer patients from which we had tumor tissue. And then separately, there were women who had hysterectomy and healthy ovaries removed in that context, but did not have any cancer. So we used that as healthy control tissue. Everything else is the same. Here just gives, they were all high-grade serous ovarian cancers. Here are any indications for the healthy, but you can see none of those indications are ovarian cancer related. And so here shows the results. In this case, all were correctly classified, but overall the model tends to have less confidence here than in the other 2 sets of results, but still accurate classification. Here the model weights. Oh, and I should have mentioned, in the case of ovarian cancer, when we used 4 mirrors, we didn't get that, the results were not that exciting. So we tried again using 5 mirrors and got good results, which is not surprising given that in that crystallographic structure analysis, we did see that there were CDR3 sequences for which more than 4 amino acids were in direct contact with peptide. So in this case, we needed to use 5 mirrors in order to get good classification. And then this just shows the 3 sets of biophysicochemical patterns in comparison, and they are all distinct. So our primary conclusions were that tumor-associated T-cell receptor repertoires contain T-cell receptors with biophysicochemical signatures that can reliably distinguish them from repertoires associated with healthy tissue of the same organ type, and that these patterns appear to be specific to the cancer type. So our next steps, first, most important, is to validate on unseen data. So even though we did leave one out, cross-validation, that's, does not constitute validation. So we're in the process of sequencing separate patient cohorts in order to evaluate the model performance. And then also we're teaming up with experimentalists to do, because my lab is only computational, to do experiments that will assess whether the receptors containing these patterns have any relevant reactivity. So I don't know how I'm doing on time. Okay. So in the case of multiple sclerosis, I wanted to show this because it's B cell receptors rather than T cell receptors. So that's kind of interesting. In this case, what we had was we had two sets of patients. And again, these are UT Southwestern patients. We had patients with multiple sclerosis, and then we had patients who were seen in clinic for some other neurological disease that is clinically difficult to distinguish from multiple sclerosis. And we had cerebrospinal fluid from both sets of patients and B cell receptor repertoires from those cerebrospinal fluid B cells. In this case, we did not have anything like crystallographic structures to drive our choice of motif length. So we just empirically tried, I think, size 4 up to size 7. And we observed that a length of 6, so 6 mers, gave the best results in this case. Also, because of the way the sequencing was done, we weren't able to use any information about receptor relative abundance. So this model was just based purely on Ashley factors for each motif. And so here's the OND patients, other neurological disease and relapsing remitting multiple sclerosis patients. And we also had originally just worked with amino acid sequence. And you can see when we switched to Ashley factors, the classification accuracy improved. It's much lower than what we saw in the cancer cases. I don't know whether that has to do with B cell receptor antigen interactions, and then maybe this is harder, or whether it could be because we didn't have the receptor abundance data available, because we did see in the T cell receptor case that when we ran models excluding the receptor abundance feature in the model, the performance was not as good. But again, we see that the disease case for which we're attempting to predict labels, all are correctly classified. But in the case of the controls, we have some that are incorrectly classified. And this is the biochemical motif that the model is using as a flag. Here I'm showing the CDR3 sequences for each of the patients. These are just the RRMS patients. So I should add to this the 3 OND patients who get a high score, but I didn't think to do that. But again, you can see, as in the case of the T cell receptors, that the amino acid sequences do differ between the motifs. So the model is picking up on their physicochemical properties. Now, in this case, I should have mentioned for all 4 cases now that I've described, we did do permutation analysis, and in all cases, over 100 permutations, so not a large number because these things run for a long time, but over about 100 permutations, the average classification accuracy was around 49%, and there was no observation of a classification accuracy that was near as large as what we observed on our real data. But in the case of MS, we were fortunate to have an unseen cohort, so a completely separate cohort of patients that weren't used at all for model training. In this case, we had 102 patients, and you can see the classification accuracy does drop when we move to unseen data, but it's still better than clinicians do in the clinic. Sadly, for this disease, it's very difficult to diagnose. And yeah, that's it. So this work was done, so you see Scott right there. This work was done as the postdoctoral project for Jared Osmeyer, who's shown here. Here, my collaborators, Nancy Munson, and I should have a picture of Ben Greenberg. So they were primaries on the multiple sclerosis study. For the cancer studies, Jasmine Tiro, Jayanthi Leah, Elena Lucas, a clinical pathologist, an oncologist, and a cancer prevention and control scientist. And this work was supported by NIAID, CPRIT in Texas, and 2 local private charities. And with that, I'll happy to take any questions.
Thanks very much, Lindsay. So questions, we've got time for plenty of questions.
Hello, Lindsay. So, in your work to discriminate the cancer tissue and the normal tissue study, you say that it seems to be the cancer specific. And what do you think is a biological reason for this cancer specificity? This is my first question. Yeah. And the other one is that you said you are trying to validate the different cancer types in additional samples data, right? So did you try to do it in breast cancer, right, besides the one you did? Because I know that there are several other data sets available for the breast cancer.
Yeah, so far we haven't attempted to validate in any of the 3 cancer data sets, and we are working to do sequencing on local UT Southwestern cancer patients for colorectal and ovarian cancer. We wanted to do breast cancer also, again, on local UT Southwestern patients, but it's just a bandwidth issue. We just haven't gotten that rolled into the queue yet, but we do intend to do that. I wasn't aware of other publicly available data sets that might be appropriate for us to use as validation, so maybe you can point those out to me. That would be maybe a better next step for us. Regarding biologically what's driving it, that's a really interesting question. So one possibility, I guess, is that there's some shared tumor antigen that all of the patients have an immune response against. That seems maybe unlikely, even though there is evidence of shared antigens across patients of the same cancer type and with different cancer types. So not neoantigens, obviously, but dysregulated genes that are, overexpressed, for example, cancer testis antigens in these. But they tend, even though they are shared across patients, they're not usually present in every patient with a particular cancer type. So I don't think that all of these patients would have those, but that's something we could test for using other omics technologies, and that we haven't done. Another possibility that we thought about is there's a lot of tissue damage. And so you're probably getting autoreactive kinds of immune responses or just cleanup immune responses where there might be some tissue-specific antigens, antigens that are present in healthy tissue of that type but aren't usually exposed or for which you don't usually have an immune response. And so we may be picking up on those responses as well. And maybe people in the audience have better ideas. But those were the 2 things that we had thought about.
Hi. Here.
Can you wave? Oh, thank you.
That was a really nice talk. I just have a couple of questions. The first one is that in case of false positives, like in the healthy patients being misclassified, I was wondering if you saw any pattern in their Ashley factors. And the second question is, in the case of where you're using 4-mers for certain cancers, did you get a chance to check how the 5-mers would perform?
Thank you. Yeah. So regarding the 5-mers, as I mentioned, those do better for ovarian cancer. We never tried those for breast and colorectal cancer. We are working on another approach now that will allow for gaps. So it doesn't have to be, a 4 or 5-mer strictly speaking, but will allow for gaps. We tried that also on ovarian cancer. And in that case, the parameters in the model that specify where the gaps are located were really tiny, suggesting that the model isn't really utilizing that information. But that may just be in the ovarian cancer case where it doesn't need that. And if we try in other disease types, it may be that ends up being an important thing. To the first question you asked, remind me what your first question was. Sorry.
Thank you so much. Like in case of false positives,
Oh,
I was just wondering what the Ashley factors of the healthy patients look like.
Yeah, good question. So the high scoring CDR3 sequences would contain and do contain snippets with the appropriate biochemical pattern, and that's why they receive a high score. I should have included those on my slide though, so you could see what the amino acid sequences are, and I neglected to do that. But you do have the same biochemical properties, snippets with the same biochemical properties. Thank you.
Any more questions?
I was just thinking about, this would be, I guess, bulk TCR sequencing from these sections, and if the signatures you're finding could be confounded by the presence of TCRs from Tregs, for example, so rather than finding...
Definitely, these could be Tregs that we're picking up for sure.
So it could be a blend of immunogenic versus tolerizing, or one or either.
Right, exactly. So all we can say so far is that these motifs are present and they appear to be uniquely present in patients with the particular disease type, but we don't yet know anything about why they're present and what they're doing. So we hope to disentangle that with follow-up experiments. And of course, we would love to get, so this was archived tissue, and so we would love to get fresh tumor tissue where we can do flow sorting or single cell where we have both chains as well as some information about phenotype. So those would all be really interesting.
So Lindsay.
Yeah.
Great talk, really
Thank
hard
you.
to do, and it's amazing what you're doing.
Thanks.
So is there a correlation that you see between the reliability of the classifier and the clone rank?
So I'm not sure I'm completely understanding your question, but in the case of MS, we don't have clone rank information, sadly, because of the way the sequencing was done. In the case of breast cancer, we were tending to pick up on the largest clones in the patients for the most part. In the case of colorectal cancer, we were not, interestingly. Some of them were very low, but for both models, we had similar classification accuracies. I think it was 93% for colorectal cancer and 94% for breast cancer. I don't know if that's really answering your question, though.
No, it's just interesting. Actually, it is. You see here, it looks like some of the clones that are lower ranked discriminate well, but some of the ones that are low ranked don't.
Right.
So
like here, this one is basically 1.
Right.
And this one is also basically 1. So, yeah.
Any further questions? Okay, well, will you thank Nicholas and Lindsay? And before we break for strong coffee and Italian cakes, Pam would just like to have a word.
Great. Let's thank them first.